So, hi everybody, thanks Anna for the introduction. I'm Maria, one of the curators in the GWAS Catalog team, together with Elliot, who will help me with the Q&A. We will try to make easy and entertaining this presentation. Thanks for joining and so your interest in the catalog. So the GWAS catalog is quite straightforward to use. So we are gonna focus in doing a brief introduction to key terms in GWAS. Later on, explain why it's important, the aims of the GWAS catalog, how we structure the data. We'll move to, in practice, data catalog access, how you can access to all the data in the website. We explain the data submission, and in the end, if you still have more questions or you want to discuss more things, we'll have a few minutes for the Q&A. So first of all, and before I forgot, if you have questions in the middle of the presentation, please don't hesitate to start to type in, and we will try to manage to reply your questions. So first, what's the genome-wide association study? This methodology relies on the concept that for complex diseases or phenotypes, there might be more than multiple variants in the genome that affects the likelihood of an individual to have certain traits along with environmental factors. Another important concept is that maybe you are familiar that usually they are saying that GWAS is an approach well-established, hypothesis-free, and that's because it's across the whole genome. It scans the whole genome in comparison with fine mapping or gene analysis that is based in previously published results. Another important thing to keep in mind is that the SNP associations are not maybe are not the causal variant because of the LD plots. So what appears in the catalog or in the studies is those SNPs that were genotyped and were with those p-values, but conclude that those are the causal variants, further analysis is needed. Another thing that you need to know is how GWAS is designed. So usually have two stages, discovery and replication. For the discovery, you have an initial sample, which is genotyped by at least 100,000 SNPs, or maybe several millions, because the genotype technology developed quite a lot in the last years, and can maybe drawn from general population or for a continuous trait like BMI, or can be split in cases and controls if the trait is a binary case. All the samples can belong to one ancestry or may be drawn in different ancestries. Later on, the most significant SNPs can be replicated in an independent sample that can be from the same initial ancestry or another independent one. All the samples can have one common trait or can have also a background trait. And later on, I will explain more in detail what does it mean. And as I mentioned before, we need to keep in mind that to conclude that there is the causal variant, you need functional studies carried out to properly say that that's the causality of and the disease mechanisms. So how do you perform GWAS? The individuals are genotyped across the genome using different technologies, like the SNP arrays, or more recently the sequencing-based ones, like whole genome sequencing or whole exome sequence. The data then is analyzed to find the variants or haplotypes with a higher frequency in cases than in controls. Cases should be the individuals with the trait and the controls without the trait. Later on, a statistical analysis is carried out to indicate how likely is that the difference that you find in the frequency in both groups is due to an association with that trait or not. The statistical significance is the probability that the observed difference between both groups is due to a chance. So if the p-value is really low, means that it's not because of chance, it's linked to the trait. There are two key pieces for data from GWAS. One is the p-value, that is the calculation of the significance of the difference in frequency between cases and controls, and the effect size, that is the magnitude of the difference between both groups. So which kind of phenotypes can appear in the GWAS catalog, because people study them in the GWAS studies? Complex phenotypes, not Mendelian traits, which are caused by multiple genes in combination with environmental and lifestyle factors, can be common disease like type 2 diabetes, or also rare ones like frontotemporal dementia. Can be also endophenotypes like biomarker levels, which may be applied to different disorders, but almost can be anything. In the latest count, over 4,000 traits are recorded in the GWAS catalog, and you can see that can go from the common diseases to coffee consumption or political preference. Why do we need the GWAS catalog? So the aim of the catalog is to provide summary standard data from published studies. Right now, at this point, there is no a common standard way to report GWAS, the different study designs, different ways to represent the data, the criteria for replication. Sometimes the GWAS data is not clear, is in supplementary data. Sometimes how the data is presented is not straightforward. So what we do in the GWAS catalog is make the life of the users, researchers easier, showing everything in a standard way. We then make everything freely accessible and searchable through our website. And we also do two things. So from the published data, we extract the top SNPs, but we also host the full genome-wide data sets, which we call summary statistics. We later on explain more about that, who can be submitted to our repository and can be also be freely accessible through our website. Sorry. So the aim of, sorry, I have a problem now here. Our users, the goal that our users can, once we curate all the data, we basically have the SNP A is associated with the trait B with an effect size and significance C, which has been demonstrated in population D of individuals. And thanks to that, our users can do all these different things like prioritize candidate loci, do statistics on the disease data and research, prioritize for target discovery, understand the disease mechanisms, and predict the disease risk with PGS. Common questions, frequently asked questions by our users. The catalog can be used if your question is what's the best candidate loci for my functional study? Have any of my associations have been previously published, or what I found in my study is new? Are these previously known associations that previous associations in the catalog fail in my data? Or which of the SNPs that I found in my data are linked with other phenotypes? As I mentioned before, another thing that we started to do a few years ago is host also the summary statistics. Those are the full data set that has been analyzed in a study, not only the top SNPs. The utility in the latest times is quite big, and for that reason, a lot of our users are requesting summary stats. With the association data for all the variants, you can make easier the identification of collocated signals across the genomes, increase the potential downstream from meta-analysis, do new polygenic risk score analysis, integrate the summary stats with other omics like GWAS, EWAS, PHIWAS. You also can combine the summary statistics from different traits and trying to find the pleiotropy, estimate the SNP heritability thanks to that, or other analysis is the Mendelian randomization. It's also important to let you know that the GWAS catalog use and is incorporating many tools and databases. So even if you don't use directly the GWAS catalog, it's useful to understand where the data come from and how we structure it. So the next point is how we focusing in the associations. First, we need to decide if a study is eligible to be included in the catalog. For that reason, to ensure the consistency, we follow strict criteria. The study needs to be genome-wide. The technology, at the beginning of the story of the catalog, we were extracting arrays-based studies. We extended the scope in 2017 to targeted array. And since this year, we are starting to extract the data from sequencing studies, independently of its its exon sequence or whole genome sequence. Another criteria we follow is the study needs to include at least 100,000 SNPs analyzed and need to be published. For the SNPs we extract in the catalog, the SNPs need to be with a p-value below 1.-5 in the overall population, and we extract the most significant SNPs for its independent locus. If you want to know more detail, you can read more in the documentation we have in that link. So the data that we include in the catalog, for each one of the publications, we can have one or multiple GWAS because the publication analyze different traits. And for each one of the GWASs, we have the experimental and sample metadata. So what do we mean with experimental and sample metadata? Each study in the catalog contains study design information like the sample size and the ancestral description. To extract information and present in the catalog, we do in two forms. With the free text that is following what the authors describes, is flexible to include several experimental design details that we will need that could be useful for the users, like cases and controls or whatever is appropriate for the study. And we also extract like in a structure sample description, which follows control terms that increase the consistency and makes easier to do a search, and is also machine readable. What this is mean? So, for example, in this study, if we see the sample description of the cohort is not easygoing, it's quite descriptive. So we can summarize in the free text, as you can see here. We have the number of cases and number of controls. In the structure way, we saw the following a broad category that could be easily findable and searchable also for machines. The broad ancestries we have are these ones, and if you are interested in know more about this part of the catalog, we published in 2018 a paper about this ancestry framework and all the details about how we do it. More data that we include in the catalog is linked to the genotyping method and also the number of SNPs analyzed in the study. And finally, we include the association results. So when it comes from the association results for each one of the SNPs, what we extract is for the variant, the SNP identifier, the effect allele, the gene, and the position. The mapping is done through the ensemble. Later on, we extract the association, as I mentioned before, the p-value, which is the strength of this association. And later on, the measure of that effect size could be a beta or odd ratio. The beta is in case that the trait analyzed is a quantitative one through logistic regression, like BMI. And the odd ratio is for the qualitative traits when we have cases, for example, cases and controls. And other thing that we extract is the trait. The trait is extracted also in two ways, the specific reported trait and the ontology. This is similar to what I explained for ancestry. So we have the reported trait similar to the free text trait description, follows what reflects what the authors have been published, whereas the structured text follows an ontology. In our case, most of our ontology follows the experimental factor ontology. In case you are asking what's an ontology, it's a set of terms, and the relations is like a family tree of traits, includes all the synonyms for that trait and the relations with other traits. So, for example, if we think in ice creams, for sure each one of us will think in one of them, different shapes, different flavors, different things. So the ontology help us to structure all our ideas of ice cream in a structured way. In another example applied to GWAS Catalog, for example, for chronic lymphocytic leukemia, here you have all the synonyms for this term, but also the relations it has in the family tree. So we know that belong is one type of leukemia, which is a subtype of skeletal system disease, but is also one kind of immune system disease, or could be a chronic disease, or could be a genetic disorder, or could be a cancer. So EFO help us. Other thing that I mentioned before is what happened with the background traits. That's something that we improved this year in the catalog. In the past, when we several times we have that the authors want to do an analysis in one trait, but the individuals also have another trait. For example, in this case, allergic rhinitis in asthma. So the authors have not directly interest in study asthma, but all their samples have asthma. So we improve how we represent this in the catalog. In the past, the trait was allergic rhinitis in asthma, and the main trait was all together, whereas now you can when you are in the website, you can select that you don't want to include in your analysis those background traits. So you would be focused in the previous case in which individuals have the allergic rhinitis. And right now, I'm not sure if I'm going too quickly. We can move to the data access. How we can search in the website based on different things. The first of them will be the publication or study. The second one will be focusing traits, and we will see the locus sum plot tool. Other thing that we can search in the website is by variant, and we will see the LD plot tool. Sequence by gene. I will mention also how you can access to the summary statistics, how you can download the data and access programmatically. So for this, I need to switch off from here and open this part. Okay, so if you go to the catalog, the main web page, if you click in the search, always show us the latest association publications that have been added to the catalog. This is not something that maybe is really useful, but is one of the options. If you type whatever in the catalog, you will see 5 the different things that we can search from. Again, maybe it's not really useful. So as I mentioned, we are going to start to search by publication. So you can search by publication based on if you know the name of the author, or if you know the trait that appears in the text. But the most what we recommend is that you search by PubMed ID. So here I search by this PubMed ID, which I have already saved. And I select this PubMed ID because I wanted to show you that in the catalog with this symbol explain to you that there are summary statistics available, and this other symbol say that this study, this publication was based on targeted array genotypes. So if we go to this publication, we can start to see all the details for this PubMed ID, where it has been published, the authors, which one of them has the ORCID ID. Also explain that there is full summary statistics available. You have links to external resources like PubMed and European PMC. And if we start to scroll down, first we will have the associations that appears in this study, in this publication. And in this case, this publication have 3 different studies. So first, you have the associations, or you can order based on traits, based in genes. You can also order based in which one is the top SNP, the one which has more significant p-value. And if we continue scrolling down, you have the 3 studies. So as you can see here, for this publication, we have 3 different reported traits, each one of them with different metadata associated. And one of them has also summary statistics. So if we go to that publication, that study, we have all the details, all the metadata associated to that study. So in this case, it's a clear example of what I explained before about the free test. We have different subtypes of thyroid disease. So we have the cases for one of the subtypes, the number of cases for the other subtypes, and the number of controls. And in the structure ancestry, we have the broad category that are Europeans. We also have access to the summary statistics through the FTP or through the API. And if we scroll down, we have all the associations that appear in this study. Something that that is useful to keep in mind is that all our tables are customizable, and you can select which headers you want to maintain, the information that you want, and later on you can download in a CSV format. Okay, so the next thing that we can search, we did publication and study. The next one could be traits. So you can do BMI, you can do, I don't know, breast cancer, or the coffee consumption. I select asthma. And you will have all the traits similar in the ontology. The first one is the one which is more accurate to our term. This will take a little bit to load. Here you can see that for this trait, we have other external resources. OLS and OXO are 2 external resources for ontologies. So you can see how this trait is located in the tree. Okay, we can wait. We have also linked to Open Targets, which is a resource for drug discovery, and you can have access to the GWAS Catalog. As mentioned with the background traits, here you can click or not click if you want to include also the background traits. So each time that asthma was not the main trait to be studied, but is present in the samples. Again, if you scroll down, you can see the associations. You can customize the headers, the columns that you are interested to see and download, and you can download. If we continue scrolling down, you can see the different studies that have been included in the catalog for this trait, and we also could see. For example, you can filter here if you are interested in all the studies that have been published for a specific ancestry, and you can download only those studies. If we continue scrolling down, we found the locus sum plot. Here is a representation of all the associations across the genome that have been published in the GWAS catalog. If you click in each one of the dots, you can see all the information for that SNP with the PubMed ID, the author, the publication, the p-value. Okay. The next thing that we can search for is based in if you found an SNP that you are interested in. So you can, if you know the RSID, you do the search, and in this case, we will give you as return the SNP and also the gene where it's located. So if we go to the variant page for this SNP, we will see the location is based, as I explained at the beginning. All our mapping is follow ensemble pipeline. So here you have access to the ensemble main pages, but also other resources like dbSNP and also UCSC. If we started to scroll down, we have again all the different tables for this SNP. This SNP has been published 22 times in 22 studies for 12 traits. So if we started to scroll down, you can see all the times that this SNP has been published, in which studies, and also the different traits. As you can see here, has been published 6 times for platelet count, but also for other traits, usually linked to blood cells. And if we continue scrolling down, we have another tool that is the LD plot. In this case, it's showing you the LD of this SNP linked to in relation with other SNPs published in the catalog or in ensemble. Depending on the shape, you can see the variants published in the GWAS catalog, or you can see the variants published in ensemble. The colors are linked to the trait category. And other thing that you can start to customize is the variant of reference. And if you change the population of reference, if you change the population, you can see that the LD changes. You also can customize the values of the LD. And later on, you can download what was your preference. So if we go back to do another search, we did publication, we did studies, we did traits, we did variants. So another thing that we can do is search by gene. So I select FOXP2. you can see here the opposite. You can see that there is one gene, and also you can see all the variants that are mapped into this gene into the in the GWAS catalog. So if we open this gene, we have again all the mapping data linked to the NCBI, the information of this gene in NCBI, in Ensemble, in Open Targets, and also in OMEM. Again, you have the different tables that you can customize. The studies and the different traits. I mentioned before, as one of the external links is to other resources is Open Targets, but I also wanted to mention another resource, which is a sister catalog to us, which is the BGS. Part of the team are working on BGS. So in case you are interested in polygenic risk scores, both catalogs are synchronized. So for this publication, you can see all the things that have been extracted as in BGS. But maybe you do not want to do a search in this way. You want to see all the data that has been published in the catalog. So for that, we offer you two options. So one is download all the data. So you go to downloads. You can download several spreadsheets with all the information. That can be the associations, all the studies, or all the ancestry, but also for the studies which has not been published yet. Those with summary statistics that the users submit and they want to make available to the community before the publication is released. And another option that you can have is to have access to all the data, but in another way, which is through documentation and the API. The API is the programmatic access to the catalog. So if you are a bioinformatician or you have a bioinformatician in the lab, is quite useful to if in case you want to do some searches. For example, you have a long list of variants that you want to pull data out from, or if you want to do more complicated combinations that are not easily done in a search and filter with the spreadsheets. The next thing I wanted to speak you that we offer in the GWAS catalog is the summary statistics. So we have the available studies, all the available studies with summary stats. This page maybe is gonna take a little bit to load. I had it open before. Okay, I had it open here. So if you go to this page, we offer you two different kind of tables. One is the list of all the published studies with summary stats. The second one is the list of pre-published or unpublished studies with summary stats. But we also provide the community all other resources because not all the summary stats are publicly available. So we are aware of that, and we show to the users other resources where they can find more data or the source, and later on they need to contact with the consortia or the owners of that data. So for these two tables, again, you have a you can customize the table. And here you can search by any of the things that you are interested. If you know the PubMed ID, but you are not sure it has been with summary stats, you can find here doing the search. You can download everything in CSV, and here you can have access to the FTP or through the API. Through the API, we are currently going through the upload to all the data. Right now it's around 50% of the summary statistics, but you can have access to all the summary stats. So far, right now you can see it's more than 22,000 summary statistics from published studies and more than 6,700 for unpublished. So you can access to all of that through the FTP. So if we click the first of them, you can see two links. One of them is the raw data that has been submitted, that was available on intranet or the author submits, and the other one is in a folder called Harmonize. What Harmonize means? What means is that we edited this data to make the meta-analysis for users more straightforward. So here you have two files. The first of them, one of them is with edited headers. So all the headers have the same name, whereas the second one plus have the same standardized headers is also mapped and orientate to the forward strand to the field of interest. Okay. So I think that we can go back to the end of the presentation, if I can. So the next thing that I wanted to mention was the data submission. So for the data submission, you can submit full summary stats for variants that have been genotyped by array, by targeted array, but also by whole genome. And we will, you can also extract the associations and submit that to the catalog. And another thing is that you can submit your summary statistics when the publication is still not published, and you can access to an embargo if it's your interest, or say to us when you want to make the data available or make available. As you can see, it's more than 6,700 data sets have been published in the catalog with from publications which are not indexed in PubMed yet. All the data need to fulfill a standard format. To submit the data, you just need to go to our website and click in submit and follow all the steps. To understand all the steps, you can go and see a presentation made by Elliot last year. So for that, you just need to go to documentation and related resources, and you can have access to this presentation when all the steps are explained. And if you want to have help from us, you can contact us through Twitter in GWAS Catalog, where we announce the date of release, information, if something happened with the catalog, new data and interesting things. You can subscribe to GWAS Announce, or email us in case you need help because you are looking for something and does not appear, or you don't understand, or you disagree with our extraction. If you want to submit and you have problems, you just need to go to GWAS subs, and we will be more than happy to help you. And yeah, if you want to see more webinars, you can go to the webpage from EBI training, and as Anna mentioned at the beginning, there will be a survey for this webinar. So if you have questions, now I'll stop sharing.
Hi, Maria. So I think there have been many questions, and Elliot's been very busy answering lots of them. Before the two of you have a look at the ones that are left over, I just wanted to highlight one that he'd already answered, that asked whether the GWAS Catalog is free to access. And obviously just wanted to highlight that it is free to access, and you can go there and explore the data as you wish anytime.
Okay, the first question from Salvatore: to set a notification system to know when a specific catalog is available. So we update the catalog if everything goes well every 2 weeks or once a month, because we are all the time including new data. We prioritize the data that we include in the catalog. Lately is to include those data submitted by authors, or somebody asks us for a specific study. So it's what we prioritize. The submissions, if the study has summary statistics, or if has been requested by author. But yeah, if you want to receive notifications, you will have one every 2 weeks, more or less. Have cDNA or pDNA for protein and no. What we extract is what is present in the studies. And so later on, if you want to go to do further analysis, you need to access to other resources like EMBL to have access to or other resources at EBI for proteins. How we can filter GWAS data for African population only. So you can, as you mentioned there, you can do by publications, and when I was showing to you also by traits. In the tables, you have different headers, so you can filter there and have the table and customize that table and download that table. But if you want to have access to all the data in the catalog, maybe it's easier and better doing programmatically. Similar catalog for plants. You are not the first person who asks about this. Unfortunately, I'm not aware of. I know that there is an ensemble for plants at EBI. I know that there is interest for GWAS, but I don't know a catalog right now. Maybe in the future we can speak about another sister catalog. Can we download a table with calculated LD? Yeah, in the LD plot you can download the data. And you can customize what do you want to, which values are for you. So by default, there is a value, but you can customize that and download that data. It's possible to download from the command line on Unix. Yeah, if you do programmatic access from the data, you can integrate. Well, I'm replying everything. I don't, because Elliot, you reply a lot of questions.
I can jump in maybe. I think maybe the closest to that would be to use the API.
Yeah.
So I answered to another question about the API and gave the link to the documentation. So maybe that's a good place to look to find out how you might be able to do that.
Mm-hmm. The next question is, what should be the significant population size for GWAS and how many times it needs to be replicated? Can we only choose the varying genotype for replication data? Interesting question. So depending on the trait and depending on the type of population, the effect size is gonna be different, and unfortunately not all the time the authors provide the effect size. So the more the merrier. It's like with ancestry. We try to ask when we have a submission to ask the authors to provide as much information as possible. The minimum population size, we don't know. It depends on the trait, and it depends on the cohort. In the community, what has been always acceptable as significant was 1-8, but the catalog includes everything, the top SNPs with the threshold that is lower. So we are including more than the community is usually including in other meta-analysis. Sometimes when a study is based in previously knowledge, what they consider is those SNPs that have been with the other threshold, whereas if you use the catalog to do your meta-analysis, you will have more data. When you search a gene, does it include if it's a promoter regulatory? No, for that, again, if you want to do downstream analysis for that kind of analysis, one source could be the BEP from example, the variant effect predictor, but currently we do not include that information in the catalog. Sorry.
Maria, I was just thinking, another thing that question might be about is if you search a gene, does it include all of the variants that are in also the promoter of that gene, not just within the coding sequence of the gene?
No, currently is the mapping, is the gene itself.
That's right, yeah. And you can check our FAQs for more details about exactly how we map variants to genes. There's a bit more detail there.
Can data downloaded from the GWAS catalog hosted on other sites, another platform? I don't know what's EMBL. So all our data is freely accessible, and in fact is used by multiple researchers, but also by private companies. So yeah, I think that can be integrated. Other thing is, for example, for Ancestry, there is another research which takes all our data and analyze and publish the results, all the Ancestry that you have present in the catalog, because it's something that currently we are not doing. So yeah, I think that if you have more detail about that, yeah, contact us in case you have any problem, because yeah, we know our data, as you can see, you can download all the data in a spreadsheet, and so yeah, it's really accessible. Regarding the current pandemic, how has this data set assisted at all? Oh, okay. So we are integrated in the COVID-19 data portal at EBI. So all the data linked to COVID, all the GWASs linked to COVID have been prioritized and included in the data portal. And yeah, again, depending on the authors, if they want to prioritize, but as soon as we have it, we publish, and it's accessible for everybody. And so we know that there was a huge interest in the availability. The catalog is for human GWAS. There was another question about plants. Yeah, for now it's only human, and there is a lot of data. So I don't know if we have the capacity to move to other, at this point, to other species. I checked a list of 70 genes. I don't really understand what's the question. There are a list of 70 genes, and the majority are associated with blood.
I think it's just that sometimes a gene or a variant within a gene might be associated in a GWAS with a particular trait. That doesn't mean that link has been really solidly established by other methods necessarily, and that might be why it doesn't come up as an annotated function in gene ontology or other pathways.
Yeah. So what we include in the catalog is what the authors published. Again, we do not include all the functional analysis. We do not go further in saying this has been confirmed. That's something that other resources do, and they refer to the GWAS catalog as like the other resources or other resources around. How strict is GWAS Catalog to only list high quality? Okay, no, we include all the publications that have been indexed in PubMed. So we do not include only the high quality. Can look like that several times because there are a lot of Nature and Science and scientific report and this kind of studies, but it's because those studies have summary statistics, have been requested by authors or by users. So that's the way that we prioritize, but we curated all the data that has been published and indexed in PubMed. And if there is some study that you think is missing, please contact us and let us know. And will be priority because it's a user request. What's the difference between GWAS Catalog and other database, GenotypeAt? GenotypeAt, I think that goes further in the downstream analysis. We take the information from the publications and further analysis is made by other resources.